microbiology has never been as near to the fore of the general public consciousness as it has been in recent years. Some areas that have traditionally been the focus of particular attention are more relevant than ever. These relate to, for example, situations where microbes contribute to disease of food spoilage. In the former case, the availability of early updates relating to the recent major events such as the Ebola outbreak in Africa, the emergence of Zika virus in South America, or the Shiga toxin producing Escherichia coli outbreak in Germany in 2011 have had a major impact. In other traditional areas of microbiology, such as the application of microbes for commercial use by the food, agriculture, and pharma industries, the developments have had a predictably lower profile but are nonetheless important. So, what is entirely novel is the way in which advances in molecular biology, next-generation sequencing, and bioinformatics in particular have reinvigorated several microbiology-associated fields of research, including those listed above, and lead to the emergence of several new areas of development. Teaching resources of microbiology. Professional societies, microbiology educators, have the responsibility to teach varied audiences at institutions that have diverse missions. The educators in these arenas are served by a vast array of professional organizations. Some organizations, like the National Association of Biology Teachers and the National Science Teachers Association, serve a broader membership. Other professional organizations, such as the American Society for Microbiology, or ASM, the Society of Industrial Microbiology, the Society for General Microbiology, the Society of Applied Microbiology, Canadian Society for Microbiologists, and Australian Society for Microbiology have interest in promoting education in microbiology, especially the Chemical Heritage Foundation also has material including microbiology and biotechnology in its resource library for educators. Among these organizations, the American Society for Microbiology is a preeminent leader in the area of promoting and supporting microbiology education at all levels. In this article, any appearance of bias toward the ASM is not intentional. Rather, it is a reflection of the ASM's dominance and preeminence in this area. In fact, professional societies in other subdisciplines of biology often consult the ASM for advice about educational resources and support. In addition, the American Biological Safety Association is specialized in the area of laboratory safety and standards. These have been updated to include first response to microbial species that have the potential to be bioterror agents. In the United Kingdom, the Microbiology in Schools Advisory Committee, or MISAC, was founded in 1969 by organizations including the British Mycological Society consortium of local education authorities for the Prevention and Science Services, Institute of Biology, Society for Applied Microbiology, Society for General Microbiology, and Scottish Schools Equipment Research Centre. Their website has extensive resources for education at all levels. They sponsor meetings, competitions, and help to promote. Forensic microbiology is an old science in a new application. Its introduction to the forensic sciences has been a necessary response to terrorist threats. The benefits that microbiology brings to criminal investigations extend well outside those required for the investigation of terrorism. In return, microbiology has benefited from the enormous advance imposed by the need to develop tools for the purpose of this article is to describe how microbiology is applied in the investigation of bioterrorism, highlighting the modern advances in technology, particularly the DNA technologies, which have assisted this discipline as a forensic practice. Properties of probiotic bacteria. Microbiology is an ancient biological science in which identification and characterization of microorganisms and their interactions with the surrounding environment are studied. Recently, genomics, transcriptomics, and proteomics studies were used in this traditional field and induced much progress. In fact, the current use of genomic data in combination with mass spectrometry technologies for the analysis of microbiota, proteomy is opening new interesting fields in microbiology. According to the articles of topics that have been reviewed and discussed in this chapter, it can be concluded that proteomic techniques in both gel-based and gel-free techniques can be used as diagnostic, differential laboratory, and prognostic tools in microbiology and probiotic studies. However, these techniques need to continually progress in both basis and application to find appropriate situations in the microbiological studies. In my opinion, this field, proteomic in microbiology, especially in probiotic benefits effects, evaluation is novel and needs more research and researchers. Also, due to the importance of proteins as the final functional molecules in all of reactions, the obtained results from these studies can open a new world for scientists. History of microbiology. Microbiology as a field is defined by the size range of its objects and interest, thus relies on both instrumentation and interpretation of the unseen. The history of microbiology is one of the linked advances in laboratory technology and biological insights. The disparate organisms, their ecology, biology, chemistry, and physiology, as well as their genetic mechanisms, have provided key insights into the areas of science, including medicine, agriculture, fermentations, metabolism, food processing, and genetics. The interplay of this insight has had a major role in the progress of biological thought over the past several centuries. Introduction to pharmaceutical microbiology. Microbiology is a biological science involved with the study of microscopic organisms Microbiology is made up of several subdisciplines including bacteriology, the study of bacteria, mycology, the study of fungi, phycology, the study of algae, parasitology, the study of parasites,